All right, so I think in the interest of time, we're gonna get started. Thank you for making the hike. I think it's gonna be a really interesting session. We have some fantastic speakers. So a couple of housekeeping items. We're gonna have some time after each talk for questions, and then we're gonna try to maintain a little bit of a buffer for the end in case you have questions that came after the fact, and you want to have another piece of advice or guidance from the speakers. So the session today, What in the Omics, is co-chaired by Dr. Strug and myself, and it was the brainchild of one of our. Well, I wanted to say something, but I'm going to say something else. A brainchild of one of our highly respected CF researchers, Ron Rubinstein, who's here looking at his cell phone. But it's timely because as CF researchers, we've gone from looking at slides by histology to doing single cell RNA sequencing, in 20 years. So I think a lot of the field is now driven by access to technologies that flood us with data, and we have to design our studies properly, and we have to make sense of the different omics, like understand what type of information we can get from them and whether we can integrate them with clinical data. And takes a village. It takes basic scientists and biostatisticians and clinicians and molecular biologists to work together. So I think it's a really timely question to ask. What in the omics? So the big four omics are looking essentially at the central dogma of biology plus one. So you're looking at the DNA by genomics, and you're looking at the RNA by transcriptomics, and the proteins by proteomics. And then we also can interrogate the small molecules by metabolomics. What's interesting also is that we know the DNA is, is modified. Its access is regulated by a number of factors. So you can look at the epigenome. We know also that RNA is modified too. So you can look at the epitranscriptome. And we know the proteins are modified post-transcriptionally. So we need to also look at the epiproteome. In addition to that, we can look at how these different omics data sets interact with one another, right? In this little table, you can see that you can look, for example, at DNA-RNA interactome or RNA-RNA. You can obviously look at host and microbe data from the same sets of samples. So when you start to look at this picture, you understand how complicated, but at the same time potentially transformational access to these data can be. So how do we apply these omics to CF research? As we were thinking about this session, we wanted to help people who really haven't had that much exposure to omics to think about multiple types of strategies and applications that they can think about when they're looking to address mechanisms of CF disease pathophysiology, but also for those of you who are more interested in developing treatments and also monitoring patients. How can we look at patient samples to bring about new sets of data and a better understanding, let's say even go after non-invasive samples and understand in a very dynamic fashion what's happening to our patients. And obviously, we want to close the loop once we figured out new mechanisms, and we know when the disease is, metastable or changing. Can we apply new treatments in a quasi-personalized or even completely personalized way, and look for efficacy of those treatments using the same methods? So what we'll do today is we'll review the recent advances in both cell and fluid profiling, and also it's important to talk about the limitations of those omics methods. They cannot do everything for us, and we're limited by access to patient samples. And really the sense that we can make out of these data. So it's important for you all to challenge our speakers with regards to research implementation if you have questions that pertain to your own research. And these are the learning objectives that probably attracted you to do the hike and come to this room. So I won't go over this. We have fantastic speakers today. Dr. Karpiewicz is going to be first, and she's going to talk about statistical and design considerations when analyzing and also planning for a use of omics methods. Dr. Chandler, who's a dear friend and colleague of mine from Emory, is going to talk about multi-omic approaches and just hopefully integrating some insights from both cell and fluid assays. Dr. O'Neill from UNC Chapel Hill is a seasoned researcher in the field of GWAS and other fields, but we asked her specifically to talk about the identification of candidate genes and how those can lead to better insight into mechanisms of disease. And then Dr. Stanton, who has been a pioneer in the field as well, will be talking about the application of omics methods when it comes to predicting and monitoring treatment responses. So we're going to just get started now with our first speaker. Yulia, if you can come to the podium, please. Floor is yours, and the button here is a little hard, but
yes, thank you.
Thank you.
Hello, everyone. Can see some of you. Thank you for the introduction, and you saved me one of my slides that I now don't have to describe too much. Thanks, everyone, for being here. This is going to be an awesome workshop. Let's see if I can operate the mouse. I don't have anything to disclose. So the omics data, this is the slide I will skip now because for the most part, Robin has introduced this. One thing that I didn't want to mention is I don't see awfully a lot of epigenetic studies in CF quite yet. There's been a few, but far in between. So epigenetics, I think, is where we can improve what we do in cystic fibrosis studies. So the way we collect omics data is usually by the use of the top two platforms that I list there. It's either sequencing or mass spectrometry. Now, the protein arrays I added here because as a statistician, I think they can be useful in certain situations and can save a lot of variation. Now, the sequencing is usually done using the following three platforms. Illumina is by far the most widely used sequencing platform. It produces very short reads, about 150 to 150 base pairs, and then bioinformaticians go in and, as a puzzle, put it back together and assemble a genome. Now, PacBio and Nanopore are two sequencing platforms that allow us to obtain very long reads. Now, 10,000 base pairs is just a dramatic example I wanted to put there. In reality, your read length will depend on your pre-processing steps. For example, if you're using PCR amplification, your reads are going to be a little bit shorter because, well, it takes time to amplify longer reads. So my approach to long read sequencing, and I'm a very big proponent of long read sequencing, guys, is to use rapid sequencing kits that allow us to get from sample collection from the participant to sequencing within a couple of hours without chopping up the DNA into short reads. Now, both Nanopore and PacBio provide us with the long reads, but they do sequencing slightly different. One elongates and creates the complementary strain. The other one pushes a single strand through the nanopore, and the changes as each base goes through, the changes in current are detected. Now, just for, again, dramatic effect, I wanted to put a picture of PacBio sequencer and a nanopore sequencer next to each other. Now, one can sit on your benchtop. As a matter of fact, we have three. PacBio is usually limited to centers unless you have a lot more money than we do. Mouse problems. Here's how nanopore sequencer looks like, one of them. And moving on to mass spectrometry, the instrument in the middle is something that can sit on your bench top. It does take the entire bench. What the processing of proteins, in this example, I'm showing you proteome experiment in mass spectrometry. We still purify, just as we purify RNA and DNA for sequencing, we will purify proteins or metabolites. In the experiments that involve proteins, we need to digest the proteins into peptides and then ionize and subject them to liquid chromatography and ionization to produce something that I'm showing as those spectra on the right top. And then again, bioinformatics approaches allow us to match those spectra to actual protein, or in this case, peptide sequencers. Now, the reason I'm showing you a picture of the dotted plots on the bottom left corner is because it shows you one protein with 6 peptides. Peptides are shown on x-axis, numbered 1 through 6, while the abundances are shown on y-axis. And what I want you to get out of this is the peptide abundances come from the same protein and they're different. Yeah? So there is a bit of variation, and possibly protein arrays can actually eliminate that variation and the need to perform peptide level protein abundances for me as a statistician, because protein arrays will measure the intact protein abundance. All right, so now to finally get into the current challenges that I can identify in our research. So first of all, that's phenotypic diversity. We humans, unless you're working with inbred mice, we humans are much more phenotypically diverse, and that usually comes from either our genetic material or our environment or the interaction of the two. As such, we will address all the solutions in a little bit. The next challenge is high dimensionality. What we mean by high dimensionality here is the number of omic features, whether it's genes or proteins, metabolites or lipids that we are observing or measuring. We get a very large number of those features versus we usually, especially in CF, have fewer number of participants. Which further brings us to the next challenge, and that is we always have to perform multiple tests. What I mean here is for each gene, we're going to perform a test that says, is this gene differentially expressed between treatment groups? And we do the next and the next and the next gene, protein, metabolite, whatever we're measuring. So that is multiple testing. And the reason it matters is because when we do a statistical test, we usually pick a p-value cutoff of 0.05. And what that usually means for us is that we're happy to accept a chance of making a mistake that is about 5%, right? So when we perform multiple tests, that changes and it's no longer 5%. And we don't necessarily exactly know what it is. So that situation can be further exacerbated by confounding variables that will have a relationship between, they have a relationship to your predictor variables as well as your outcome, your gene expression or protein expression that you're measuring. So now that we have the problems, we do not despair. We can address all those issues. Apologies. So how to address phenotypic diversity? One way to address it is to use stratified analysis. So we can make sure that different phenotypic groups are well represented in our study population, and we further include that as a variable in our analysis models. We can further incorporate environmental data. Maybe somebody living in humid environment versus dry environment would have differential disease progression. And we can definitely use multi-level or cluster correlated designs. For example, if we want to study the nutritional effects and we provide nutrition to one child, what can happen is if the rest of the family decide to go to McDonald's, then that child may do what? audience participation. Well, the child may skip the meal we provide, right, and eat McDonald's. It can get even worse if we have multiple people involved in the study in the same family, cohabitating family. If we deliver different food to them, and there's been studies that done that, and then they look at each other's food and eat each other's food, and we just spend a lot of money and we get nothing out of it, right? All right, so cluster randomized design would randomize the entire family to the food, and that means if you have a lot of money, also deliver food to families that are not involved in the study. That will eliminate them from going to McDonald's. All right, next, before we start collecting omics data, so we know, I'm just making sure I'm online here. Before we start running sequencing or running instrumentation, whichever platform we use, we need to think about what run order we run our samples. So I'll give you another example. If I tell you today I ran all of my cases and I plan to run all of my controls 6 months from now, what would you say? Bad. Okay, very good. I agree with you guys. Really bad experimental design, because I have now confounded my runtime with my biology that I'm really, really interested in. And as a statistician, I no longer can decipher the two, right? Because they were run at different times. So what do? We're going to randomize our sample order, right? Now, we can't always completely randomize our samples. I have one study that's undergoing now where our participants have not been born yet. But some of them have been born and swabbed, and now we can sequence their data. So in that case, what we're going to do is we are going to run samples in batches. And within the batches, we're still going to randomize our samples, and we're still going to think about stratification. Maybe we will match our participants by X and Y chromosome. Maybe we'll match them by age. Does that make sense? So each batch will be randomized, and we're going to wait for the rest of our participants to be born. All right, so now that we have collected our data, there's still a few issues we need to be aware of. And that is that multiple testing issue of performing a statistical test for many, many, many genes, many proteins, etc., any omics feature. So the traditional method that I'm listing here is to perform our statistical test, adjusting for whatever covariates we put in the model. Then we take the p-values and we adjust them using one of the adjustment methods. Now, Bonferroni is not quite usable in thousands and thousands of comparisons, but there are some methods that are widely used, and Benjamini, Hodgberg, and Story's Q-value are probably among the most widely used. Now, the point of doing an adjustment is if you think about your, let's say, 10,000 proteins, the p-value distribution, if you were to plot them, would have a peak at 0 because there are some differentially expressed proteins, and the tail going towards 1, which is highly not differentially expressed. And the adjustment would simply shift your distribution of p-values from 0 towards 1, just a little bit. And that's the idea of multiple testing adjustment. Now, adjusting for confounders in every model, and that means in that test that we do first before adjusting, if that confounder is not necessarily linked to the outcome, maybe losing some statistical power that doesn't need to be lost if we can simply eliminate that from the model. So that's one of the things that's been solved in this proposed 2D FDR. So this method was only proposed a couple of years ago, and they proposed this two-step procedure. That's the 2D part of the FDR. They screen out the irrelevant features using a model that does not include a particular confounder. They call it an unadjusted model. And then they further drill down into only now the subset of features where they look for that true signal for the differential expression. Now, this proposed method only worked for normally distributed outcomes. So for example, in protein expression, we usually get a continuous value, and we can get the distribution of abundances to be approximately normally distributed via some transformation, for example, log2. But we sometimes get count data or binary data. So count data, for example, can be produced by RNA-seq, where we count the number of transcripts for each gene. So technically, that is not normally distributed data because it is not quite continuous. So another issue that was brought up by the authors is that type 1 error inflation could still be present if we apply this method, if the confounding has a very strong effect. The same group then about a month ago published a paper where they improved on their method, and it now accommodates additional outcomes. So your outcome can be now binary in addition to normal as well as count. So this pretty much covers all of our omics data that we're collecting. They claim, they do improve power by screening out some of the features where we do not need to use all of the confounding variables in the model. And this provides a unified approach to approximate the joint distribution of the two tests that they do, one with the unadjusted model and one with the adjusted model. Now, as a statistician, I highly appreciate not having to come up or determine what the joint distribution is. That can be quite complicated. And you can enjoy the formulas in those papers. Now, the only complication I see with this particular method is that the authors decided to not make it as an R package, but instead made it into a GitHub repository. So it's all available, but just a tiny little bit less usable than if they made it into an R package. We're all familiar how easy it is to install and use R packages. So if anybody, please suggest to the authors to do that. All right. And that actually is my last slide. I come from Perth, Western Australia. I work for Telephone Kids Institute, and we're home to the reasonably well-known RSCF, the Australian Respiratory Early Surveillance Team for Cystic Fibrosis, and that's where a wealth of my knowledge comes from. Thank you, and questions?
Thank you very much. So feel free to come up to the microphone, ask a question, or to do it online. While we're waiting, maybe I'll go first. So you mentioned that maybe epigenetics is understudied in CF, and I know, say for example, from some of the long read technologies, you can now get methylation data. So what do you see the role for the methylation that comes out of the long read sequencing in CF?
Yeah, absolutely. Thank you. I love this question. I wanted to note, and you brought up a very good point. So if you're doing long read sequencing, you're sequencing the DNA and just simply taking that same sample and subjecting it to a different bioinformatics pipeline, you get DNA methylation. So you don't need to sequence a second time. Once you sequence the DNA, you get the DNA itself as well as all of the modifications. So it's a really cool way to get your methylome. Now, the reason the methylome matters is because in humans, cytosine methylation is very important. And usually if you methylate the promoter of a gene, you will silence it. It's not 100% guaranteed. And if you demethylate the promoter, you will activate that gene. So basically, the methylation experiments can be very parallel to transcriptomics, or the data that we get from transcriptomics, and correlation is usually in the 70s to 80s percentiles. I see a lot of value in looking at host pathogen interactions. So for example, in our center, we obtain bronchoalveolar lavages of sputum from participants, and we can subject that to DNA sequencing and get host DNA and pathogen DNA all sequenced. So we can decipher what microbiome they have and the host response to that microbiome by simply sequencing the host DNA. Now, a lot of the times the host DNA is depleted so that it doesn't compromise, or it allows for more of the microbiome to be sequenced. But the value I see is if we do use long-read sequencing, then we will get the host response, and we can build the networks between microbiome, what I call is a microbiome host interactions. Thank you.
So, couple questions from somebody who reads these grants all the time.
Thank you. I need to talk to you.
When you're looking at these data sets, what sort of technical replication makes you feel comfortable, as well as what sort of sample sizes make you feel comfortable?
Yeah, so someone who gets a lot of rejections on my grants, take this with a grain of salt. Yeah, so we recently submitted a grant application where we looked at 88 participants, and we have shown the percentages and differentially expressed genes that we were able to obtain, subjected it to PASS. PASS is a statistical software package that allows us to estimate sample sizes. So our preliminary data was pretty large. 88 participants is actually considered large. We looked at the levels of differential expression, and in our follow-up experiments, we're aiming at 400 participants, but we're satisfied with about 280 being sufficient sample size. Now that still will vary, that will still depend on so this is transcriptomics, and you may need to think about variations you will see in metabolites and other things. So some preliminary data is essential for us, obviously, to decide where we're going to draw the line.
And then technical replication.
Yeah, technical replication. As a statistician, I would love to have technical replication. As a person who applies for grants and doesn't get them, I don't have money to run technical replicates. So what we try to do, and is exactly what I outlined there, is we try to make sure that we run our samples, we batch and randomize them, so that technical replication becomes less essential. Now, in mass spectrometry, it's actually very common because mass spec usually is not run in parallel, right? It's one sample at a time. So that's when we actually almost always put some quality control samples, and we monitor over time. So that differs between sequencing because we can multiplex when we sequence, so we can multiplex our entire batch. In mass spectrometry, absolutely essential, and maybe Joshua can tell us more about that. But we usually include, so Pacific Northwest National Lab, the largest mass spec facility on the planet where I used to be a while back, has a QC sample within beginning and end of every batch so that any variations can be normalized. So yeah, QC samples in that respect come in as technical replicates and are very good to be able to normalize anything. But in sequencing, we do very little replication, unfortunately. And I'm not sure if I can justify. It costs a lot of money, especially PacBio will cost you an arm and a leg. Nanopore will cost you a little bit less. Illumina will give you short reads. But I'd like to talk to you more about that.
Thank you, Yulia.
Thank you.
We'll have a little bit more time, hopefully at the end of the session, if you guys have other questions for Dr. Karpiewicz. And then we're gonna switch over to Dr. Chandler from Emory. Dr. Chandler is a power user of Omics methods, so he's gonna give us the user and scientist experience. Joshua, forward and backwards.
Yeah, thank you. Thanks for being, and thank you all for being here. I'm going to indeed talk to you about multi-omic approaches to discern complex phenotypes in cystic fibrosis. I have no relevant disclosures to tell you about. A quick outline of my talk. I'm going to address some challenges that I think are lingering in CF. I don't think this will be controversial. I'm going to do some refresher on omics and multi-omics. Rabin and Yulia have already set me up beautifully, so I'll speed through that. I'll talk about a seminal project called the IPOP cohort as an introduction to multi-omics done at a really high dimensional scale and done really well. And I'm going to talk about some advances in CF for multi-omics that really go after a couple of different axes, groups of related publications that I hope you'll appreciate along with me, and some conclusions and future directions. Okay, so challenges lingering in CF. We actually, a lot of us were just at the plenary too, and this was really well addressed there, set this up beautifully as well. But in short, there are a number of things that remain challenging for CF patients in the post highly effective modulator era. This is just one kind of prototypical example of a subject who was enrolled in a trial for ivacaftor. There's a period of her pulmonary function being masked, and then you can see pulmonary function gain, but there's still a declination. So the modulators are not necessarily removing lung function declination entirely. Furthermore, other versions of this similar result have been presented, but there's not an ablation of infection in the post-modulator era either. In fact, in some cases, infections rebound. And what's more, inflammatory mediators such as proteases may not be addressed at all by the modulators. And so it's really incumbent on us as researchers to work on what nuanced pieces of the puzzle still remain in CF. And the position I'm going to be taking today is that omics research and multi-omics research is really the way to get at that. But I also want to make a point on this slide that we don't just get to take for granted that we're going to get these samples. We not only hear about sputum being dried up, but we also have to think about the fact that anytime we ask somebody to show up to the clinic and donate a sample, or even donate a sample at home potentially, there is a burden being imposed, even if the person is very happy to be involved. And as patients get healthier, the perceived benefits, I should say, of research may actually be decreasing in the minds of some of these individuals. And so we as researchers, again, have to do our best to maximize data output and really refine our research to be very accurate and very high content. We have a lot of potential for mechanistic elucidation, biomarker production, and hopefully minimize specimen consumption so we can do more per study visit. And again, the position I'm taking is that omics is the perfect answer to this challenge. So I think we've already heard from both of the prior speakers, Rabin and Yulia, about the different omics domains. I did want to introduce it a little bit as well. I'll speed through this, but I kind of think about it a little bit like MC Escher's lithograph, Three Worlds, where there's like a fish under the water, and then you can see leaves on the surface of the water, and then you can see the trees reflected in that surface. I didn't use the lithograph because it's copyrighted, but you can see here our fish is our genomic component. This is the informational domain. And then there's kind of an interface with the transcriptome where there's still information being submitted from the genome into the working components of the cell, but there's also some functional aspects of the transcriptome as well, like with non-coding RNA. Then we have the proteome that encompasses a lot of function and also is responsible for making functional and signaling and nutritional components in the metabolome and lipidome, which interact very directly with the environment, especially with the exposome, such as like what we're eating and chemicals in the environment, microbiota as well. And of course, you could also point out, you could flip the microbiome and the host in this whole scheme, right? But that being said, to put all these things together is rather challenging. And I think one roadmap for doing that is Mike Snyder's IPOP cohort, which began with actually just Mike Snyder. And he's very public about the fact that the one subject of this 14-month study was him. And basically over this period of time, he donated his PBMCs and serum. And there were a number of methods applied to this, omic after omic after omic, just pulling from the prior slides. And these were sometimes analyzed in isolation. In most cases, that was how things began, and then integrated research or analyses came later. One of the really intriguing things about this study was Mike, as anyone, encountered viruses. And so he encountered viruses during the course of the study, including a respiratory syncytial virus during which the infection of that caused his, or was at least contemporaneous with, an increase in his fasting blood glucose to a pre-diabetic level. And it turned out through looking very carefully at the genomic analyses from his study that he had several genes that were variants that predisposed him to type 2 diabetes. And so did his mother, if I recall correctly. There's this black bar that I don't really know a way to kind of highlight it, but you can see really clearly on the x-axis, starting around 370 days or so, there's a lifestyle intervention that Mike undertakes with low sugar and exercise and some other components. And after about 4 months from the time of the RSV infection, his blood glucose gets back under control. But this is kind of an example of like scientists self-directed personalized medicine, which I think is rather a landmark. You can see here from the same study during the viral infection component, the team has tried to condense as much information as possible from the metabolome, the transcriptome, and the proteome here. And it's rather intriguing to me because the pathway that comes out is maybe one you'd expect, right? We've got glucose regulation of insulin secretion, and this we just were talking about glucose control. But here's what's interesting, and this isn't hardly the first time I've seen this. If you look at the metabolites in the cluster, those metabolites are not they're not glucose products or precursors in any immediate sense, right? In fact, mostly they're fatty acids or they're related to vitamin D metabolism. And lauric acid is the only one that at the time of that publication had really had any connection at all to type 2 diabetes. And so these studies can really give us some insights into novel metabolism, novel biology, in addition to being grounded in what we understand well from things like diabetes risk and connect it back to things that our patients and really all of us are encountering. Just to quickly add as well, that the IPOP cohort continues to this day and has more recently published about I think a 30-ish patient study that includes a lot of sex differences. So if you are a female, you are more likely to have higher expression post respiratory syncytial virus exposure and other respiratory virus exposures of a number of things that you see here in pink post-infection, and then gray would be male overexpression of the thing, and more so than female participants. And this is across genes, proteins, metabolites. So I think when we think about how to take care of our patients and how to design better therapies for them, and we were just hearing about the female, the sex difference in disease as well, we need to think about these things too, because like the innate immune response was different. Okay, so I do need to keep going through the talk at a relatively quick pace, and so I'm going to move on to the cystic fibrosis component. And I'm just going to talk about two thematic areas that kind of nucleated in my mind as I was preparing for this talk, because I could see a couple of different sets of papers that sort of pointed to a couple of different themes. So one is more of an axis of inflammation that's near and dear to my heart through hypochlorous acid production, and Rabin knows this well about me. But there's more going on than just what I'm interested in out there in the literature, and especially in the multi-omic space. Additionally, there's some personalized microbiota profiling that's been done as well in a couple of different publications I'll highlight. So as far as the first set, the one study published by O'Connor et al. In 2022 looked at the bronchoalveolar lavage of 68 CF patients and 22 disease controls. And the goal here was to compare the metabolome and the microbiome and see what kind of network associations could be made in order to discriminate what's going on in CF that way. And first, just looking at the metabolome, there was a random forest model used to pick which metabolites could well discriminate CF from non-CF here. And mostly it was short-chain lipids, sphingomyelins, lyso-PCs, and you see MET-SO, that's methionine sulfoxide, so that's an oxidized form of methionine, and then we'll keep hearing about that. But these metabolites, these lipids and methionine sulfoxide were all positively correlated with things we associate with inflammation and infection, so white blood cells in the BALF, percentage of neutrophils being higher in the BALF, and total bacterial load in the BALF. This is actually replicating or agreeing with something that I published previously with Rabin and with colleagues from Erasmus University Rotterdam, as well as Emory. Lokesh Guglani, who's in the audience, was also involved. And this showed that methionine sulfoxide was correlated with neutrophils and myeloperoxidase, which is an oxidizing protein we'll talk about more. But just to say that this is something that we have seen previously, but what the O'Connor group also did is with the microbiota, looking at 16S sequencing, they found that there was a really strong relationship of methionine sulfoxide specifically to a number of amino acids, but mostly to Staphylococcus, a very strong positive correlation, and really strong negative correlations in the green lines to a lot of other bacteria that are not CF pathogens. And so methionine sulfoxide in the study ended up being this keystone where if it moved up, then all these other things like inflammation, infection, and CF pathogen went up, and vice versa. So that kind of insight tells us that we could maybe do a lot with a little from this multi-omic approach. I then want to talk about, in the same vein, a study that I was a part of, and it also involved Yulia with the Arrest CF group. And this is specifically from colleagues in Melbourne. But they set up a study of patients who either did or did not go on to develop bronchiectasis by the age of 9. And we looked retrospectively at their bronchoalveolar lavage, including metabolomic and lipidomic analyses, and compared that to conventional biomarkers to see whether we could come up with better ways to predict bronchiectasis, whether it would or would not occur. Because I don't have a lot of time, I'm going to really jump ahead to some of the key findings that definitely were crucially involving Yulia, I must say. And we found that there were 3 major clusters of metabolites and lipids from the 2 different omics that were put together here. And the first 2, we can see if they were higher, then bronchiectasis was less likely to occur when the patient got older. In the third group, they were higher when the patient was likely to develop bronchiectasis. And you can see this includes lactosyl ceramides, which are enriched in neutrophils, it turns out, and methionine sulfoxide. And so these 2, methionine sulfoxide and lactosyl ceramide, actually outperformed neutrophil elastase in the youngest patients, age 0 to 2, in terms of predicting that bronchiectasis. And so this is another important insight about how we can maybe monitor and care for patients better, especially the inflammation side. But nevertheless, I think it's also important to consider that we didn't really have a mechanism exactly for where methionine sulfoxide was coming from. And we have a good idea that lactosyl ceramide comes from neutrophil membranes, but we also need to work on that, I should say. I'm going to tell you the methionine sulfoxide story a little bit more in that my graduate student who recently, and Rabin's graduate student who recently defended and graduated, Susan Kim, she published this paper showing that we could model which oxidants were produced by myeloperoxidase from the neutrophil in an immortalized lung cell model, very kind of a primitive model, but just to see which oxidants produce which metabolites. And we found a lot of things. It's an omic experiment after all. And I will draw your attention quickly to this cluster here of methionine sulfoxide and methionine and dihydromethionine. And you notice that the kind of 3rd and 4th circles are really dark red for methionine sulfoxide and dihydromethionine. That's because hypochlorous acid generating conditions and hypobromous acid generating conditions made a lot of this. But other conditions, the other oxidants that myeloperoxidase can make don't do it. So hypothiocyanous acid, for example, does not make this, or hydrogen peroxide, the precursor molecule, doesn't do this. So if we see this molecule, it's a tip that either the halogenating oxidants must be present. And on top of that, in the CF airways, we really only think hypochlorous acid is likely to be present because of the bromide substrate being low. Excuse me, let me go back. So the kind of the conjoined model is that neutrophils are probably contributing to a lot of these signals, and there's still chicken and egg to work out, such as the Staphylococcus versus the neutrophil, what's coming first, although I think we all have our opinions about that, and I'm happy to talk about it. But I think the important thing to point out is that we also, because we know there's a role for hypochlorous acid and we know its chemistry well, this suggests some potential interventions that we could begin trying. I want to cover another set of studies. This is both from the CF rapid response, and the first is about a lethal pulmonary exacerbation. So these are much more personalized, like the IPOP study. So in this case, a CF patient, of the CF01 in the study, had several exacerbations and a large loss of lung function over their last year of life. D0 is the day of death. And you can see that their white blood cell count goes up toward the end of their life. Their lung function is declining. And several omic data points were collected during the course of this study. And what the group ended up doing is comparing the microbiota in terms of their composition and their activity with metabolomics. And so one thing that they found was that there was a really high ribosomal RNA enrichment of E. coli, and it was actually Shiga toxin producing E. coli, as well as Bacillus, whereas Pseudomonas was responsible for most of the mRNA in these data. And what else was really interesting is that, I'm going to probably mess this up, globotriaosyl ceramide, I believe, GB3, was low for the duration of the study, but this is a Shiga toxin receptor that is expressed whenever Shiga toxin is present and is actually important in its uptake into epithelial cells. And the precursor for this notably is lactosyl ceramide, which you heard about earlier in the other study. And you can see here lactosyl ceramide was already elevated for quite a long time in the patient, and then toward the end of their life goes up. And you can also see some metatranscriptomic analysis in B and C that confirms that there's a Shiga toxin expression in the patient in both subunits A and B. The same group in another paper, and focusing on another patient, had looked at microbial abundance in sputum and compared the communities and also just the transcriptional activity of those communities during an exacerbation with intensive antibiotic component, the A. In B, there's no antibiotics, it's been withdrawn. And in C, just clindamycin has been added back in. And so you can see that there's an increase in transcriptional activity during the B phase. Whenever, again, I don't really have a good pointer for you, or maybe I do, something like this. You can sort of see the Pseudomonas is increasing in transcription toward the end of B, and then during C, with clindamycin being reintroduced, goes back down. And they take an approach of looking at the metatranscriptome of expression of genes that could feed Pseudomonas via butane diol production. And so you can actually see over time the different butane diol production genes go down during the first course of antibiotics, go back up during the off phase, and then down again for all but a couple of genera less associated with the pathogenicity in C, whereas Pseudomonas's consumptive genes also go up during B and back down during C. And this leads to the kind of working hypothesis that the Pseudomonas is using its biofilm to partner with anaerobes in order to keep them fed and persist in the environment. And by targeting the anaerobes with clindamycin, the researchers are arguing that in this individual, through the personalized omic profiling, we're able to tease out the importance of that intervention. And maybe that's a lesson we can learn from in the future. So in conclusion, multi-omics research can discern complex and subtle phenotypes in patients by providing high content data, even from minimal samples. This is something we really need in the age of the until it's done age, if you will. There are still so many things to tease out, and I think this is one way to do it. Inflammation-wise, I talked to you about the importance through multi-omic studies that point to hypochlorous acid generation by neutrophils as probably being a critical component of some of the inflammatory disease progression we see, and also related to CF pathogen presence in one study. And in infection-wise, the CF rapid response team has shown a couple of times over that there's some really exciting and important nuances to the microbial communities in the CF airway that we can continue to pay attention to. Where do we go from here? I think Yulia made some fantastic points already. I would argue that we definitely need to be thinking even just about data storage next session. I mean, just my mass spectrometer in the lab generates more data than I can technically store for free most of the time, so we have to come up with crazy plans around that. But just that's important. Data sharing, optimizing studies and home monitoring, we're hearing about that. I think we need to think about the need for genetic counseling in the context of this. And of course, developing new tools, even AI, and Yulia already talked about some important statistical tools as well. So thanks for bearing with me. There's a lot of people who deserve credit for what was shown here. And I just want to say thanks to all of them, and thanks to you for listening, and take any questions you may have.
Again, if there's any questions, please come up to the microphone or put them online, and maybe I'll get started. So I'm just curious. So I thought it was really interesting how you're interpreting, multi-omics in individual patients for personalization. Yeah. Just curious if you're thinking about how to do multi-omics integration. I mean, the data so complementary, so across cohorts if you want to increase sample size.
Absolutely, yeah. I don't know if I can tab through. Okay, by the way, this is a circus plot. This is how Mike Snyder's team did that, and it's a lot of work. I often don't end up doing that, but what I can tell you about firsthand, if I can find it, here we go. There are tools that are kind of ready to use like right out of the box that you can even get on the web and use. This is XMWAS, and so I actually did this study as a postdoc. And what's nice about this is that you can construct networks. So here's how you integrate by constructing a network between omic A and omic B and whatever you might care about, such as FEV1. And then the interesting thing about the personalization question is you might go from a patient who has one kind of outcome or one kind of infection and compare that to another patient. And what this tool and a lot of other tools allow you to do is they allow you to actually compare the two networks in terms of where do the variables fall in it in terms of their centrality. So what we did here was we actually asked that question about myeloid and lymphoid cells in an influenza response, and what we found was that treating mice with cadmium actually made the myeloid response more rigorous or more robust, and not necessarily in a good way, mind you, but there were more myeloid cells in terms of the regulation of transcriptome, metabolome, et cetera. I think that those kinds of approaches could be deployed in people as well. Yeah.
Are there any other questions?
Well, Joshua, I'm going to ask you, you sort of touched upon this a little bit, the cost associated with omics studies. And you just talked about storage, mind you, okay? Not even actually running them. So can you give the audience a little bit of a, feel for maybe, and we've been working on that for years. So, what's the cost if somebody comes to you and says, well, Joshua, can you run, these metabolomics panels for me and maybe riff on that a little bit?
Yeah, absolutely. It will vary, but I think if you wanted to do the state of the art today, like in a metabolomic experiment, then I would advise you to budget for around $100 per sample, probably at a minimum, really, just because if you just add up the things like the solvent and the plastic and so on and so forth, you won't quite get there. But then what you'll find is that maintaining that instrument, having the columns for the instrument, having the personnel time to work on that, especially the personnel time. I think honestly, if anything costs like the most in this imaginary pie chart, if anything takes the most of it up, it's really the personnel time, because so much of this requires in the absence of like an AI or something, a person to really what am I trying to say? It's like they're committing what is it when people used to examine the chicken bones, like haruspicy or something like this. But essentially, you have to look and examine the chicken bones in your data set forever and ever until you finally see like this 3D image appear, and oh my gosh, it's this. It's actually very fun, but it's also expensive because that person costs time. And so I would say, yeah, if I want to put a number to it for like something like my lab would do, plan for around $100 a sample, but it will vary depending on the scope, depending on what you want to do. And thankfully, if you get functional or if you have a hypothesis going in, you can often reduce down what you want to do, right? So if my lab is saying, no, we care mostly about the pentose phosphate pathway, actually, we only need to measure lactate to even get a surrogate measure of the pentose phosphate pathway based on carbon-13 labeling of that molecule. Well, now we only have to measure really one thing in kind of two forms. So if you think smart, you can save some money. But if you want to go big, if you want to go Mike Snyder, IPOP, then it's going to be more expensive.
Well, that's why I'm happy to have Joshua in my team, because I don't have to deal with that personal cost. Although, yes, somehow. Anybody else has questions for Joshua? All right, so I think we're going to thank Joshua. Thank you.
Appreciate it.
And then we're going to welcome Dr. O'Neill to the podium. Good to go on that. Do you want me to activate the pointer?
Yes, as long as I can hear.
So it's the laser pointer here, and I think if you do this, going to be able to see it, I assume. Remember, this goes forward, this goes backwards.
All right, I'm the seasoned researcher. I haven't been introduced that way before, so I guess that's nice. All right, so I'm going to give you a history of the human GWAS and CF and talk about what has been happening throughout time with GWAS. And I don't have any disclosures to make here. So CF is a monogenic disease. So, why are we thinking about GWASs? We know that the CF gene causes cystic fibrosis. It's a high penetrant. It's well known to be recessive. And yet, what we've learned over the years that there are certain phenotypes within CF that clearly, if you have a mutation in the CF gene, you're going to experience this phenotype. So absence of vas deferens in men, high sweat chloride, the development of airway disease across a lifetime. So these phenotypes are very highly penetrant and not very variable. And then there's another group of phenotypes that are controlled or regulated by the mutation in the CF gene itself. So you can have the mild mutations or the severe mutations. And those mutations make a big difference as to whether or not you get pancreatic enzyme deficiency, meconium ileus, diabetes, and liver disease. And then there are these phenotypes that are somehow dependent on other factors, such as environmental factors or non-environmental factors, which are genetic. And one of these, of course, is the rate of lung function decline. And so what I'm trying to do is move this thing. I'll just do that. So this is what we're trying to explain. So we have the exactly same genotype in this group of Toronto patients, delta F homozygous, 841 patients. And we have FEV1s that look like this, where some patients have very, very low FEV1 very early on in life, and some patients that out to 40, 50 years have a very high lung function. And we could say this is environmental, but we know there are families like this. So here's a family, and in this family, there were 6 children born with CF, and these children died very young. The ones in the dark, they died. They were in this category. This child is 58 years old and is on modifier therapy now. So whereas these patients were, before the modifier era, delta F, they had the phenotype that was down here and they died early. There's something about this patient, this CF patient, that allowed him to survive, and now he's taking advantage of the CF modifier therapy. So within the same environment of this family, there's clearly something that protected this person, and we would like to know what this is, because, the thinking going into this kind of study is that if you could really understand what protected this person in the first place, perhaps it's a therapeutic target that would allow other people to also benefit. So that was, the thought going into it is that if you could, it would be a good thing to do if you could identify these modifiers and turn them into a therapeutic targets. So there were several studies that started out around 1988 and carried on through the 2000s that defined the heritability of some of these CF traits. And it turns out that even though there are large environmental components for some of these traits, the family and SIB studies that were conducted also demonstrated a very high proportion of genetic contribution to these phenotypes. So the idea with GWAS studies and gene candidate studies is that if you could identify the causes behind these, then again, you have a therapeutic target. So how does this work? Well, you have, for example, CF patients with low lung function versus CF patients with high lung function, or CF patients with diabetes or meconium ileus and CF patients without. And CF patients with X phenotype versus the CF patients without this phenotype. And you're looking to see if there's differences in the frequency of genetic variants between these two populations. So in this little example, it's pulled from the GWAS catalog homepage. This case frequency for this SNP is 58%, and the control's at 16%. So there's a clear difference in frequency. And so if you do that across the genome, what we're looking for are hits, and these are often depicted in what's called a Manhattan plot. I think it's because it looks like the skyline of Manhattan. So it was named after New York City. And so the hits along the chromosomes here show up as peaks with high p-values. So the significance of the finding, the association to the genetic variability shows up above the genome-wide significant line, or often people also talk about suggestive SNPs, where they're above the general background, but they don't reach the genome-wide significance level. And then once you identify these regions, then usually what happens is you show these regions kind of blown up in a locus-zoom plot. This is one of our early locus-zoom plots from our paper back in 2011, which showed the top SNP in the region between EHF and APIP on chromosome 11. So these are how these locus-zoom plots look. They show the top SNP, they show the p-value, and they give you a representation with these colors of the linkage disequilibrium around that SNP. So those are common ways that these things are depicted, and you'll see a bunch of examples of this as I go along here. So GWAS studies have been going on for a long time. Back in 2007 is when they, sort of started coming out. And over time, you can see that they've increased in number, but also importantly, they've increased in the size. And so these are for, many, many different phenotypic traits. The phenotypic traits now can be things like height or weight or educational status, cardiovascular disease, diabetes, goes on and on and on. And the cohorts are really sometimes very large, well over 100,000. And along with that, the number of participants that are participating within these GWAS trials are increasing. And as you increase your sample size, you increase your sensitivity to detect these variants. And so the number of associations is also increasing at this quite dramatic rate. So here's the current GWAS catalog that I pulled a couple of days ago. It has 6,000 publications, 537,000 variant trait associations, and 64,000 full summary statistics files. So within this are a few of our CF studies. So a very active area of interest across genetics research for quite some time. So let me give you the timeline of CF genetic modifier studies. So as I mentioned, the early studies in the twin and SIB studies that identified the high heritability traits of CF were taking place during this time frame. The first mention of GWAS in the literature was 1997, and the first person that was recruited into a candidate gene study at UNC under the direction of Mike Knowles in a collaboration with Mitch Drum at the time at Case Western was around 2000. It took 5 years to recruit the population that led to the first validated candidate gene out of our group. That was published in the New England Journal of Medicine. And then another 10 year, or how many years? 6 years before the first linkage study was done. There was a second, GWAS linkage study was done with Gary Cutting at Johns Hopkins. And then the first full GWAS was done in 2015, and then a recent one after the whole genome sequencing project, which took place from started in 2015. The sequencing took a while to make happen, and now the papers are coming out in 2023. So let me just take you through some of these studies and show you how that all looked across time. So the first thing that you need, well, one of the three things that you need in order to do these kind of studies is a phenotype that's robust and consistent across cohorts. And so there were several years where Dr. Knowles and his colleagues worked to develop the Coolidge normalized mortality adjusted lung function measure, and some details and references are provided here. And what this does is it allowed us to combine cohorts. So in the red and the blue are the mild and severe CF cohorts from UNC, combined with the Canadian cohort for the 2011 paper, and this was done in collaboration with Lisa and other colleagues, Joanna Romans, et cetera, up in Toronto. And this normalized lung function variable was necessary in order to allow for a consistent phenotype across cohorts, and it's proven to be robust and validated. Well, obviously, once you get a phenotype, then you need your genetic testing, and then you need strong biostatistical expertise in order to run these association tests. So the very first paper that was published was actually not a GWAS, it was a candidate gene study. And like I said, the recruitment started for this in 2000, and this paper was published in 2005, and you can appreciate the size of the sample, 808 people. And look what we did. We genotyped 16 polymorphisms in 10 genes, and that took 5 years. So look at how far we've come. And these were candidate genes based on the concept of known genetic variants in the literature and how they could relate to CF pathogenesis. And the gene TGF-beta, a couple of variants in the promoter region, turned out to be significant in this study. And then in 2011, we were able to conduct a full GWAS analysis with 2,494 subjects from the UNC and Toronto group. There was another study in 2015 with 6,365 CF patients represented, and the new study that was just published using the whole genome sequence data in 2023 had 7,840 patients represented. So very large numbers, a huge amount of work to get this kind of number up for a CF population, a relatively rare disease, monumental effort by the CF community to make this happen. And what you can see is what's consistently found across GWAS studies in the literature is that as you increase the sample size, you increase your power to detect genetic variants. And we went from a signal on chromosome 11 when we were using 2,000 patients to 5 signals that reached genome-wide significance at this number. And now here we've popped up to 6. So this increment is not very large. We've added another locus. And here we are with 6 genome-wide significant loci for this phenotype of severity of lung disease in CF. The other thing that happens is that the genotyping platform improves over time. So in 2011, the the ability to genotype SNPs was, I think, was about 300,000 SNPs. And so on the chromosome 11 locus, you can appreciate all the different SNPs that could be genotyped. And by 2015, the density of that increased. And here's what it looks like in 2023, where you can see, many hundreds of SNPs genotyped across this chromosome 11 region, which really gives you the ability to dig in and fine map, it's called fine mapping, I guess, these loci that you're finding in these signatures. And this kind of genotyping was especially, this density of genotyping is especially improved for other loci, such as the MUC4, MUC20 locus that was identified. You can see back in 2015, there was this big gap, which is now gone with the genome sequencing, and the same for the HLA class 2. There's a much higher density of coverage now. So along the timeline for the CF lung studies, there were also other phenotypes that were being looked at, including diabetes and meconium ileus. And as with the lung function phenotype, we were able to find, or the groups were able to find signals that reached above the genome-wide significant level, and which were improved as the number of patients increased. So as the number of patients increased and the density of the chip array was improved, the number of loci again went up in these regions. And this is the latest study from 2022 that looked at MI and diabetes from the whole genome sequence population. And one thing that you can appreciate is that look at all these loci that we have to study. And the fact that with these two phenotypes, even though they're both sort of in the GI area, or the pancreas with diabetes and MI in the intestine, the loci and genes are different, and this is a common feature. So what are we learning? One of the things that we've learned from these diabetes studies is that, you always try to learn from them, right? So the mechanism of, so what this showed us for diabetes is that the genes that are responsible for type 2 diabetes in the general population also seem to be responsible for the signatures in CF. And so this was an important implication. So it has important treatment implications. So the CF-related diabetes and the type 2 diabetes in the population, they have mechanistic overlap. And so you can think about treatments within the type 2 diabetes population that involve B-cell function would also be applicable to CF. So this is an important mechanistic study from those GWAS studies. Now the moderators wanted me to mention COPD. So I have one slide that has COPD, just to mention that studies for multiple lung phenotypes are available in the literature, from COPD to lung function itself, to asthma, to IPF. And the number of genes in these diseases that reach genome-wide significance is impressive and daunting. And interestingly enough, different. They're different. They don't overlap. Each of these diseases has its own pathology. And this is also the case for CF. So if we look at the genes, this is from a review article that came out in 2022, so it's maybe not complete, but if we look at the genes that are coming up with lung disease, meconium ileus, CF-related diabetes, and a Pseudomonas infection, they tend to be different. And there's not very much overlap. Is that me? Okay. Am I done already? Okay, all right, very good. So each phenotype is producing its own unique set, And so this is going to require us to understand these modifier loci at an organ tissue level. So when we went into this, of course, we didn't know. We didn't know. We thought maybe that we would find genes that really relate to CFTR processing to the membrane or CFTR folding. But that's, really not what we're seeing. These genes are telling us something about CFTR biology, but it tells us that, they're different in different tissues. And the other thing just to point out is that these genes that we find for lung disease severity, although they're very interesting genes in the context of CF pathophysiology, they're not the same as asthma. They're not found in the COPD. HLA is found in asthma, yes, but they're not the ones that show up high in IPF or for lung function. And so CF as a lung disease, again, has its own unique pathology that is an important message for us in the CF community, I think. Okay, and then there's also been a lot of work trying to integrate this GWAS data with gene expression data. And I don't have time to go into it all, but just to give a couple of examples, there is a method called TWAS and PredictScan, which use imputed gene expression from the EQTL data from GTACs to try to associate gene expression changes with genetic loci. And Hong Dang in our group published a paper on this in 2020. And so there's a lot of signal here. And one of the ways that I try to get my head around this signal is to put these things into pathways. And the pathways that are showing up in these signals can really be informative and part of this integrated analysis that people keep talking about to help inform us as to what these signals are telling us. So, for example, this pathway of antimicrobial peptides showed up in the genes that showed up in the TWAS analysis. And of course, when we see something like this, we say, oh yeah, it's antimicrobial peptides are clearly important in CF. And Deepika Polanini also did an analysis where she was looking at RNA sequencing data from nasal biopsies from CF patients. And her paper, which was published in 2018, has just this massive table of pathways that are significant. And there, I couldn't even fit, it would go all the way down to the floor. But if you look at these pathways, it's telling us something about what, the genetic signals are telling us what's important in CF, which types of pathogenesis pathways are important. And in our recent 2023 from the whole genome sequencing data, there was this very interesting pathway that was popping out with all these genes that were relative to, related to lung growth, development, and repair. And with all of the information that's coming out on how cells respond to stress and how they repair and how they go back in development and then reinvent themselves after infection and inflammation, it makes a lot of sense that these kind of genes were coming up. And Dr. Knoll spent hours looking and was convinced that this is a true signal in the CF lung that's telling us something that we need to pay attention to moving forward. So what now? So, I was trying to read what was going on in the other diseases where there's many, many papers and lots of work with huge sample size. And, most of them, if you read them, they say there's lots of loci and the mechanism of the association to disease development are still largely unknown. And so you can apply this statement to almost any disease where genetic association studies have been done. One of the things that was noticed very early is that these signals are often in between genes, not over genes. They're often in introns or intragenic, so they're in regulatory regions. And because of the strong LD that you can see, it's very difficult to identify the causal SNPs. You don't know which of the SNPs is actually affecting gene expression. And it continues to be a challenge for all diseases, and CF isn't immune to that. But having the fine mapping of the genetic loci that we got from the whole genome sequence will help. So for example, if you know all of these SNPs in this region now with the whole genome sequence, you can map them to the regulatory regions that are found now. And there's hundreds and thousands of regulatory regions and experiments and methylation studies and chromatin states and all this, where if you really, really wanted to dig into it, now the data is available, the association signals are available across the entire region. And this is just an example from Ann Harris's group where, they looked at, where are the SNPs falling relative to these regions in this paper. And that's how you have to try to get to understand these loci. And of course, there's all kinds of stuff we've already heard about trying to integrate chromatin states with QTLs, co-localizing your genetic signal with these other signals to try to point to the gene of interest. There's all kinds of data now out on where genes are expressed in cells. This is from the Cell by Gene website through Chan Zuckerberg. I just pulled up some of our genes that we see in the lung, and you can see the cell types that they're expressed in. And this information is just growing exponentially. So the action is that we need to have more research in this area for each of these genes. And we have seen publications of our lung function genes in EHF. If I look in PubMed and I ask, are there papers out there? Do we know? Are people working on these genes? Certainly some more than others. Lots of interest in SLC9A3 and CF, which is one of the modifiers that we have found, but we need more. So we still don't completely understand the mechanisms of these associations, and clearly more is needed for us to make headway. And ideally, of course, what we want is a druggable target. So I just put this up as an example. So in this paper, where they're looking at networks of genome-wide association studies, they make an interesting statement. These are examples of drug-targeting proteins of interest with GWAS-linked genes that are not found in the GWAS. So the development of these networks and pathways and hubs, even though you don't see them in the GWAS themselves, they're telling you something about the pathogenesis, and they're going to point you to targets that maybe already have drugs in the pipeline or available clinically for repurposing. And just for an example of a network that we are getting ready to publish from the liver disease phenotype. So all of these red genes are coming out of the Vegas analysis in that study. And when you run an ingenuity pathway analysis, they're pointing toward inflammation. So NF-kappa B, MAPK, ERK. So, this is sort of pointing to the now kind of upcoming thinking in CF liver disease that some of it might be driven by inflammation, and our genetic analysis would say that that's probably true. So one of the problems in CF, of course, is that we're never going to reach the sample sizes in other studies. So, the last study was here at 7,000. You can see some of these other studies are well over 100,000. It's just not possible in the CF community. It'd going to be very, very difficult to find more rare variants with higher effect size, et cetera, like some of these other studies are finding. So we're going to be limited that way. And the other thing that is a limitation of our current GWAS studies is that they're predominantly conducted on people with CF of European ancestry. And in this example, with Crohn's and ulcerative colitis, the signal that they got from European ancestry was different than Japanese, and we don't have any reason to expect that that would be any different. So one challenge for our community is to apply the knowledge we learned from the GWAS across ancestry groups. And finally, the current GWAS studies use pre-modulator phenotypes only. And how are we going to apply these methods to post-modulator phenotypes? So one of the things we're thinking about is trying to understand whether or not GWAS signals relate to the post-modulator phenotypes. And this is just a snapshot from our patients in our CF genome project of lung function response after modulator, and there's clearly patients that respond better than others. And one of the questions we have as geneticists is whether or not any of this is driven by the genetics and whether or not any of this is related to the phenotypes that, or the genotypes that we've already identified. So in conclusion, the GWAS studies for CF using pre-modulator phenotypes are providing important mechanistic insights. For example, the relationship of CF diabetes to type 2 diabetes in the population. And there's plenty of analysis opportunities that remain. I have the long-range sequencing. We have not done that on our populations. And this would be very useful for these complex regions, MUC4, MUC5AC, HLA regions. We haven't done much copy number variation analysis, and of course all this integration that's possible with these new omics. And even maybe just reanalyzing the data with the new methods that were already discussed would give us new insights. Of course, we need more functional studies. We haven't developed a therapeutic target for any of these candidate genes, but that doesn't mean that they're not therapeutic targets. We are thinking of, you could always think of new phenotypes. Early lung disease from arrest CF, some of the phenotypes there, are they driven by genetic modifiers? And then, of course, the post-modulator phenotypes that might be amenable to this kind of testing as well. So there's all kinds of data available. There's gene expression and data from multiple studies. There's GCHEX data from other consortium. There's whole genome sequence data from multiple CF cohorts. There's phenotype data through the registry. There's genomic information on chromatin states from ENCODE, and those are growing exponentially. There's summary statistics available on phenotype, genotype, and association. And the challenge for us is to continue to support the analysis and statisticians to reanalyze and integrate this data. And I'd just like to end by thanking the CF Genome Project collaborators. It's a massive effort, and this doesn't even report all of the clinical sites that recruited patients and all of the previous papers that and the work that went into that. And end by honoring Dr. Mike Knowles, who's retired this year. We miss you already, Mike. Big shoes to fill. So this symposium is presented in honor of all your work. Without you, none of this, I don't think, would have happened. So thank you.
Okay, we have some time for a couple questions. I'm happy to start it off if there are others who want to come to the mic first. Okay, so I was just wondering if we can turn the sample size question on its head a little bit. So maybe we didn't find, in many other complex traits, we needed hundreds of thousands of samples to identify loci, but in CF, we were able to identify loci with actually pretty small samples. So have you thought about a little bit about what that means about the complex genetic model in CF? Is it as complex as, say, some of these other traits? Does that sort of tell us something about the modification of disease severity?
Well, you're probably better at answering that than I am. Well, yeah, I mean, I hadn't really thought about that. We had to get to numbers of 2,000, 5,000, 6,000 to find 6 loci. Is that much different than the other complex traits?
Yeah, our effect sizes are bigger than the other complex traits,
Okay.
right? Yeah. Yeah, just wondering if we haven't really take a step back to say, what does that tell us about the model, the genetics of CF? Yeah. But I'm wondering if you thought about that.
No, I mean, what I think about is CF has its own unique pathology, and so maybe it's not too surprising, right? With a background of a loss of a gene that's very important for epithelial function, maybe the signatures would be stronger.
Hi, Vamshi Raju from University of Alabama, Birmingham. I've always been quite intrigued by these modifier genes. They show up, and then there are multiple verification of those. But do you have any thoughts on how they could be verified in, say, animal models to really confirm whether they're really influenced in a controlled condition, and in terms of being able to manipulate them for interventions, for example?
Yeah, so I wish that were an easy question. So we've spent a long time trying to figure out what's going on with MUC4. We spent a long time trying to figure out what's going on in chromosome 11. The animal models that we used were mice models bred to the beta-ENaC muco-obstructive lung disease, and you can knock out MUC4 and you don't change the phenotype. And so you're like, it doesn't make any sense, right? You have this strong genetic signal. You'd think that you'd be able to get a phenotype in the mice. So it's not going to be easy. So of course, one of the things that people want to do is understand which of the regulatory elements are the ones that are causing the effect, and to do that, like Ann Harris has done, to CRISPR knock out these elements and see what happens to the gene expression around the regions. Functional analysis of these genes in airway epithelial cultures under different conditions. So, I mean, one of the things that we're thinking is that maybe we're missing the time frame when these genetic modifiers are critical. Maybe it has to be early, which is one of the reasons we're very interested in correlating these phenotypes to the early phenotypes that they see in arrest CF. So to try to get a correlation between the SNPs there and those very early phenotypes. Maybe we're just missing the time frame, but it's not like people aren't trying.
Okay. So, of course, I never expected it to be an easy question. If it was, it would have been done by now. But on the other side, because of the complexity of CF disease and then differences in animal models versus human, but can you share any examples or insights of modifier genes being helpful in gathering more disease insights, or like even being able to manipulate from non-CF or like non-respiratory indications?
You mean in other diseases?
Yeah. So essentially, our modifier genes, can they be helpful in identifying new disease targets?
Well, no, I mean, they can. So, for example, in inflammatory bowel disease, IL-23 was pulled out of the GWAS study for Crohn's disease, and IL-23 is now a drug target that Crohn's patients, use, and it's effective in Crohn's patients. So it's not like there's no examples of GWAS signals producing druggable targets.
Yeah, that's reassuring because
Yeah.
IBD shares a lot of overlap with chronic respiratory illnesses. So thank you.
Yeah.
Thank you. Okay, our last speaker for the session is Professor Stanton. Bruce. This will move forward. Backwards.
I think I can.
You can? Okay.
That's what I was told. Okay, thanks a lot.
Sure.
Thanks both of the chairs for inviting me, and the program committee as well for inviting me today. First, before I start, I would just like to acknowledge my colleague, Tom Hampton, shown here on the slide. Tom is the director of our Dartmouth CF Bioinformatics Core, and he is really the driver of everything that I'm going to show you today, a co-author on all the papers. as well as a bunch of terrific graduate students and postdocs who I will acknowledge as we move along. Okay. That didn't work either. Yeah, the space bar didn't work either. Oh, there it goes. Okay. Thanks. I got to hit it hard. Okay, nothing to disclose. So I'm going to talk about mining public data and archive data. So the question is why? So comparing and combining multiple studies identifies reproducible results, discarding potential spurious signals from single studies, and importantly, something we've been talking about is it increases statistical robustness. In addition, there are also unreported insights in individual studies that are waiting to be discovered by researchers with the right biological knowledge. But there are challenges. The challenges of data mining and reusing data is acquiring and processing archive data sets can actually take months, and we actually know that from firsthand experience. So because of that limitation and the opportunities that exist for published and available data, is we build systems that allow researchers to extract novel insights from public data sets within just a few seconds to minutes without the need to program, which we think is a real benefit. So in my presentation, I'm going to talk about several of the programs that we've developed to data mine and answer what we think are important questions in the CF field. So our pipeline, so as I mentioned briefly, we build tools for researchers to study CF, and the tools are actually directly inspired by our faculty and our trainees at Dartmouth. And our process is basically based on the FAIR principles. That's findable, accessible, interoperable, interoperable, and reproducible. And so we assemble data sets relevant to a specific research question. We convert the complex data structures into more accessible formats. We make the data available to view in user-friendly formats. And we provide analytical tools to researchers so that they can rapidly, seconds to minutes, rather than days, weeks, or even months, to extract novel insights from public data, both published and unpublished. So I'm going to walk you through some of the programs that Tom and our colleagues have developed, and then I will give you specific examples of how that's led to wet bench experiments that, at least from our belief, has led to interesting observations in the CF field. So this is a study that Katja Kuppen, who was in our group for a long while, she works at Boehringer Ingelheim now, and it's called ScanGeo. And ScanGeo it's a published study, you can see on the bottom there, and it identifies studies where a gene or a biological pathway of interest is differentially expressed. And it includes microarray data for 20 species, including humans, CF pathogens, and other model organisms. And what I'm especially proud of Katja's work is that it's been accessed now by more than 1 million unique IP addresses, 1 million. Provides a detailed statistical analysis, figures and tables within minutes, and thankfully for me, it requires no computational experience, truly. And this is a study that actually, right at the beginning of COVID, that Tom and I and one of our pulmonologists published, and we used ScanGeo to try to understand or predict what effect COVID would have on CF patients. And we didn't really have enough data to answer all the questions. In other words, what's happening to ACE, the furin, ACE2, TMP, RSS2, and all the other components that lead to COVID infections. So what Tom actually did was he downloaded and interrogated some databases, and you can see on the lower left there, he was able to look at ACE2 in CF And to our surprise, that was one of our own RNA-seq studies. We didn't even know that the ACE2 levels had changed. And then a paper published by another group showed, that's the middle graph there, showed exactly the same thing, that ACE2 was up. And that same paper by, I'll probably mangle the name, Voisins and et al., they showed that the TMP RSS2 was actually down. And based on all of these findings with other published data in the CF field that probably many of you contributed to, we took a 50-50 guess that patients with CF would have less severe COVID symptoms. And I think that we guessed right, with the exception, I think, of lung transplants who are immunocompromised. This is another study that Zhongyu Li, who's sitting right here in the audience, and he's now at Colorado doing a postdoc, and he wrote a program that was based on microbial transcriptomics, and it was based on the observation that less than 4% of about 5,800 microbial archive data sets include sample group annotations required to assess differential gene expression. That's a lot of data sets that really weren't accessible. So what Zhongyu did was he developed this program that was able to increase the availability of data sets from 4 to 33%. And that's a pretty big percentage of a pretty big number. And what Zhongyu did with that information is he identified a gene in Pseudomonas PA3923 that was an unknown gene, unknown function, and he found that in about 50% of the studies, that gene actually correlated with genes that contribute to biofilm formation of Pseudomonas. So in the lower bar graph there, what Zhongyu did is he got a hold of a transposon library where that gene was interrupted, and he did a biofilm wet bench study, and he showed that when that gene was not expressed, that the ability of Pseudomonas went down, to make a biofilm went down. And so therefore he, using bioinformatic analysis, he was able to show that an unknown gene had that function of contributing to biofilm development. This is a study, CF-seq, done by Sam Neff here. He was actually an undergraduate at Dartmouth when he started working on this. He's now getting his PhD in data science at Princeton. And what CF-seq does, it allows users to rapidly explore gene and pathways of interest in about 128 studies in 1,300 samples of 13 CF pathogens, including Aspergillus, Candida, Pseudomonas, Staph aureus. The program can filter studies by strains, treatment, media type, and again, it requires no computational experience. And these are all published papers. So one of our graduate students at the time, Georgia Doing, shown here on the right, she did using this program, she asked a question, does co-culture with other CF pathogens influence Pseudomonas gene expression and virulence? And CF-seq facilitated the analysis. And what Georgia found was that Pseudomonas genes involved in phenazine biosynthesis are upregulated in co-culture with Candida, but not in co-culture with Staph aureus. Now, of course, you can see the two volcano plots there that she downloaded from the program. And but obviously, this is a prediction based on gene expression, so she has to do the wet bench experiments to test that hypothesis. So the program, many of the programs that Tom and our colleagues are right, are hypothesis generating that they can be followed up. This is another study by Katja that she published last year, SCAPE-ACT Plus. And what that does is allows you to examine differentially expressed genes from the 6 escape pathogens and an additional CF pathogens that are we all know about. And it develops insight and hypothesis. It provides pathway analysis, diagrams. And And similar programs exist for eukaryotes, but not for prokaryotes. So this is a real nice advancement in the slide. The images on the right just show you the flow chart, and the paper was published, as I said, last year. This is a case study that we did with the paper, and what's interesting from my point of view is that it confirms studies that were published 2 years ago describing the role of sigma factor in protecting C. diff against stressors. The program that she wrote identifies 3 KEGG pathways protecting C. diff against stressors that weren't mentioned in the original study. And we all know with these huge data sets that you end up with so much information, you spend years following it up, you write one paper, deposit the data, and then there's so much more information. And I think this is a great example of, number one, rigor and reproducibility, confirming what somebody else has shown, and 2, identifying other pathways that play a role in either pathogenesis or infection. This is a paper that's in progress right now by Lily Taub, who is actually a master's student in our bioinformatics program. And it combines the analysis capability of ESCAPE-Act Plus with the CF-seq data compendium. And it facilitates comparison of pathway activation across experimental conditions. And it includes 26 NCBI gene expression studies. And as I said, it's in development. And you can see in the lower table there some of the bacterial species that Lily is developing the program to interrogate. This is another program, or another example of the utility of this program. And this is Sharanya Sakar, who's sitting right there in the audience. She did an analysis using this ePath DASH program. And she was interested to see whether, to try to understand whether gene expression in Pseudomonas could tell you something about different gyrase inhibitors. And she found that, and I'm going to mangle the name, but cumarmycin is more efficient than ciprofloxacin in reducing biofilm formation in Pseudomonas. Yeah, she's smiling at me. She knows I mangled the word. So the obvious thing is it's a nice prediction based on data that she mined, has to follow it up with a wet bench to actually prove that that's the case. This, I think, is a really cool program. Again, this is Sam Neff. He worked with us for 2 years after he graduated from Dartmouth. And he developed this program that was just accepted like 2 weeks ago. And it's called RocketMir. And it's a translational launch pad for microRNA-based antimicrobial drug development. And shown here in the title from one of our recent papers that we published a couple years ago, LET7B, which is a human microRNA in vesicles secreted by human airway epithelial cells, it actually reduces biofilm formation and increases antibiotic sensitivity of Pseudomonas. And it does that by targeting many, many genes in both the biofilm formation pathway and in the antibiotic resistance efflux pump. So it doesn't target just one gene, it targets multiple genes in these 2 pathways. So what Sam thought is, well, let's develop a program, RocketMir, to identify human microRNAs with potential antimicrobial and antibiofilm activity against 24 antibiotic-resistant CF pathogens. And the program actually confirmed that LET7B and other microRNAs are predicted to inhibit virulence in biofilm gene pathways in Pseudomonas, but really importantly is that it also predicts that it will target genes in B. cepacia, Prevotella, Strep, and Staph. So that paper is in press now. I think he just did the proofs. And that this slide basically just shows that RocketMir predicts that numerous microRNA targets genes on 20 different pathogens. And this is sort of a diagram on the left that shows all the different microRNAs that you can find in the program that do target. On the right, the figure shows the number of human microRNAs that target at least 10% of genes by the pathogens. For example, on the far left in that bar graph, Aspergillus has a high number of genes that are predicted to be targeted by microRNAs, and just for reference, we put Pseudomonas there. So again, Sharanya is doing studies now and following up on that, where she's taken mesenchymal stromal cell extracellular vesicles and she's engineering them to contain LET7B to reduce Pseudomonas in a mouse model of infection. And ultimately, we're going to look at other pathogens as well. And one of the reasons that she's using the MSC EVs is that MSCs in their EVs are known to reduce inflammation, which, as we know, is up in CF and causes quite a bit of damage. And she's also found that several other microRNAs have been identified that she can use to inhibit biofilm formation. And the graph on the right is a mouse study that she did in collaboration with Tom Kelly at Case Western, where she instilled in a Pseudomonas model of infection the MSC EVs control, or MSC EVs, and they express very low levels of LET7B, but when she bioengineered the MSCs to contain LET7, you can see on the far right that there's a dramatic reduction in the colony forming units, so it reduced the infection, and you can talk to her more about that in poster 17. We're also working on another program called Respire. That's a gene expression metadata for 85 organisms in over 72,000 samples. It's 16S amplicon sequencing and shotgun microbiome data for over 50,000 samples that are now being loaded. And it includes proteomic, or will include proteomic and metabolomic modules. So to summarize, we think that by comparing and combining multiple studies of big data, we can identify reproducible results, discarding spurious signals from single studies, potentially at least, and certainly increase statistical robustness. Importantly, we think there are unreported, as we showed, insights in individual studies that are waiting to be uncovered by researchers with the right biological knowledge. Acquiring and processing archive data sets can take months, and some of them have taken 4 or 5 or 6 months of full-time work by our students and our trainees to download that data to be accessible by a click of a button. And so that really facilitates the time. So we build systems that allow researchers and CF to extract novel insights from public data within a few seconds or minutes without the need to program. So the other thing that we do is, thanks to, again, Tom, who had this idea, is we've developed a bioinformatics data mining educational program where we teach a 2-week course every summer up in Maine at Mount Desert Island, right next to Acadia Park, National Park, which is a lot of fun. And many of our trainees, or also TAs, are now giving lectures in the course. And for example, Zhang Yu, who's sitting right there, is a, and just tip the hat, he's also helped with the course design, which is fabulous. And then Sam Neff, who, as I mentioned, is a PhD student now in data science at Princeton, actually does a newsletter on data reuse and research. It comes up approximately once, or about once every month or 2 months, which not only covers CF, but it covers a bunch of other fields as well. So we're really proud of Sam and all the people that I talked to you about. And here's the group here. Current lab members, I mentioned most of them already: Tom, Katya, Sam Neff, Zhang Yu. Tom Kelly played a really critical role in helping us with some of the mouse studies, and our colleagues at Dartmouth, Deb Hogan, George O'Toole, and of course our funding, which was really critical. to all of this. So questions, we're enthusiastic to add data on bacterial strains, organism of interest to the CF community. So we'll take suggestions and requests, either email me or you'll probably get further if you email Tom, because he's really the driver of this program. And I'll stop there, and thanks everybody for coming.
Thank you, Bruce. Questions from the audience? Questions online, maybe, Lisa? Have you been able to see anything? Dr. O'Neill?
So how often do you think we need to go back to our old data and reanalyze it with the new methods? Because like you said, it takes 5 years to publish a paper. You publish it, you feel like you're done with it. But you know that new methods and statistical analysis could improve your findings. I mean, where does it end, and when is it worth it to go back? And how would you know that it's worth it to go back? Any thoughts on that?
Yeah, thanks, Wanda. I mean, I think part of it is that more and more studies are archived. It's important to upload that or download that data to add to the compendium. A lot of the things that happen, like when Tom and LA, Sharon, and I, right at the beginning of COVID, when you couldn't go into the lab, we were sort of saying, well, are CF patients going to have worse outcomes with viruses based on the fact that they usually do? Or will it be less severe? And as we were talking about it and going through the literature, there wasn't any data on ACE or the other receptor. And so Tom actually sat down and used the program and found it. So in many cases, it's going back is driven by, well, there's no data, and those data were never published in a paper, but they were archived. So I think if you have specific questions, you can use that data to generate hypotheses rather than going back and sort of, oh, I have an idea, let's do an experiment. There's a lot of data that tell us something, but we just don't know it because we haven't analyzed it. We just did it, Zhang Yu did a paper that we're working on now, and I think he found something like 20 pathways. And we pursued one in the wet bench experiments, and that took years of wet bench work. And what about the other 19 or so pathways, right? So I think it depends on your question, and you can go back. And many times, people have already done the experiment that you might be thinking about doing, and the data are there, but you didn't know it. And I think for people who live in places where there isn't a lot of research money for doing these big data experiments that, as previous speakers noted, are quite expensive. Other people have done the experiments. You can sit in a computer in a room and interrogate the data and come up with hypotheses and instigate or initiate a lot of really interesting things. Did that answer your question? I guess not.
No, I mean, I know it's a hard question.
Yeah.
It's just, like when TWAS and PredictScan came out, it seemed like it was worth it to reanalyze the genetic data that we had at the time, because that's imputating the gene expression onto that.
Yep.
It was like an advance. Now, would this new method of FDR2+, would we find new signals that we had never seen before that are going to lead us to that therapeutic target that's missing or not? I mean, it's just really hard to know because it's a lot of work to go back.
Yeah. One of our postdocs did a single-cell-seq on immune cells, and it was published about 5 years ago when single-cell sort of just started. And as soon as she thought she had an answer, another paper would come out with a new method, and she'd go back and reanalyze with the new method. And if my recollection is correct, I think what Britton found was that there was a core number of observations that were consistently identified by, the first generation, the second generation, and the third generation. And what's really interesting about mining data from 10 different labs or 1,300 different data sets is different investigators, sometimes different methods, grow the bacteria in different media, and you find the same result. And I think that indicates to me, at least, that it's probably a real biological signal, and it's not dependent on the media that you grew it on, or the specific investigator, or batch effects, or any of the other things that previous speakers talked about. So I think in our group, we really believe that using multiple programs, going back and looking at multiple data sets, can tell you a lot about rigor and reproducibility. And if 10 studies all show you the same thing using all these different methods, it's probably right. But that's just my opinion.
You sort of answered half my question by talking about the multiple data sets.
Okay.
Do you ever set up the programs to randomly select 5 data sets to come up with an observation and then replicate in another 5 data sets or whatever, kind of like the GWASs will take one population and then try to replicate in a separate population as just another way to try to get...
Zhang Yu, they're both shaking their heads.
That's an interesting thought. I mean, I think what you're trying to get at is like, what is the sort of null distribution of all these results? But I mean, so I won't totally, this is an interesting idea, but if you have a specific hypothesis, and we're talking about FDR corrections and all this stuff, the space that you're searching in is very different now. Like all the probabilities are really different. So if you're talking about ACE2, it's different from talking about all the genes. Mm-hmm. So, you're in a nicer statistical situation than just casting a 20,000 multiple test net.
And I think maybe a partial answer to that is, Zhang Yu, when you did the study on, I think it's 2923, it's something like you found about half, maybe about 40% of the studies, that gene did correlate with other known biofilm genes that went up, but in the other studies, it didn't correlate. So it wasn't like 100% of the studies agreed with it. And that's why, we got the transposon library and actually did the wet bench experiment and found it when, you took 2 different knockouts of that gene, you got the same, or the disruption of the gene, you got the same result. You get the wet bench result that half the studies predicted.
Right.
So, full transparency, we have done some follow-ups on the predictions.
Sure.
And it doesn't follow. But I would say most of the time,
Surprisingly good.
yeah, it's surprisingly good. And that has to do, I think, with the issue that you mentioned earlier, statistical robustness. If it's just our lab doing a study on our favorite cells, maybe it's right, maybe it's wrong. But if 10 other labs do similar studies, I mean, maybe if you did 100 measures, looked at 100 parameters.
It's the same thing as trying to see the same result in 2 different cell lines when you're doing a model system.
Right.
If you see it in 2 different cell lines, chances are better that it's right as
Right.
opposed to just
Yeah.
one.
But I think
Exactly.
trying to be a little philosophical about it. I think during the COVID experience that we all had, it was the first time that a lot of people were generating a lot of omics data at the same time, and within a week you could actually go ahead and figure something out, out of data mining.
Right.
That was immediately relevant to your work. So I think because we're moving towards this as a field, not just in CF, which is a monogenic disease, but we're met with a new virus. We don't know how it works, and we're going to have to figure it out. I think we should train our students on prior data sets, maybe maybe that's a way to reanalyze those data sets. Maybe not you, Wanda, but maybe your students. You put them for the first 2 or 3 months to reanalyze those data sets, and they'll learn the ropes, and they might find something new with the new algorithms, but they should at least validate against what's been found before. But I think, I mean, it's really an exciting era of biomedicine to be in, right? And all of us seasoned researchers are learning how to best use it. But I think our trainees better be, good at it, because we need them to be.
And that's one of the reasons that we teach that course in the summer. It's a 2-week course,
Yep.
runs like 8-9 hours a day. But I think the students get a lot out of it. And I'll just add one quick thing. We've actually found a lot of data sets that were never published, but they were uploaded and they were archived. And the programs that Tom and the rest of us in the group have developed actually find those data sets. And, I think all of us have had the experience where you do experiments and it kind of doesn't work, but you generate all this massive amount of data. And, there's no journal that's going to publish negative results or no results. But there's a lot of data in there that if you mine the data, you might find something really interesting. It's like the old, you only find what you can see under the streetlight, and there's a lot of other things in the darkness.
Well, that's a nice way to close the session.
It is getting dark.
It is getting dark. Thank you very much to the speakers. Thank you all for coming here and attending the session with us.